Assalamu alaikum students. Up to now we were talking about the fatty acids. This is the alternate view of the fatty acid, like in the zigzag manner, showing the carbon chain, the saturated and unsaturated ones. Now we will talk about the triglycerides. Triglycerides is the combined term for fats and oils. And they contain glycerol along with 3 fatty acids, glycerol plus 3 fatty acids, and they are connected by ester bonds. Like here you can see glycerol. This is 3 carbon containing alcohol, and with the alcohol, all the OH, one of the fatty acid is connected through ester bond. There are 3 ester bonds here in the structure. The following now may help you to remember the components of the triglyceride. Triglyceride contains fatty acids and glycerol, the simple triglycerides. Here you see the chemical composition. This is the the structure of the glycerol, and this is the fatty acid structure we already discussed in the last slides. This is the fatty acid, saturated and unsaturated both. When this fatty acid it combines with the glycerol through ester linkage, triglyceride is formed. Again, there are 3 molecules of fatty acids, and after combination they produce a triglyceride. The word tri is coming from the 3 fatty acids that combine through 3 ester linkages. Fat or oil, it depends on the melting point or liquid at room temperature. The difference between fats and oil is their appearance. It depends mostly on the degree of unsaturation or saturation. Animal fats are unsaturated ones, however plant oils are mostly unsaturated ones. Unsaturated fatty acid, their melting point is very low. However, fatty acid content in percentage by weight, and this is the trend in case of the butter. They have the higher content of saturated fatty acid. However, egg, it has the highest content of unsaturated fatty acids in it. Similarly, for the vegetable oils, soybean, olive, corn, sunflower and canola. Canola has the highest content of unsaturated fatty acid. It has a high content of oil in it. Dietary fatty acids, saturated fats associated with heart diseases like ghee or butter, they are saturated fats. They promote high level of blood cholesterol. Animal fats also contain cholesterol. Plants have no cholesterol or very less cholesterol in them. Chemical properties of triglycerides is a very important topic, and triglycerides mean the structure carrying 3 glycerols and 3 fatty acids linked through ester linkages. When they react with the iodine, they are used to quantitate unsaturation in fats. When a fat that is having unsaturation in it, it is treated with I2, then I is added in the structure, and this is the quantitative test that how many double bonds are there. Product is colorless, I2 is colored, I2 is normally violet, and this product is colorless. Iodine number, grams iodine reacting with 100 gram fat. This is the term for determining the iodine number. Iodine number, it gives the degree of unsaturation in fats or triglycerides. Hydrolysis of esters, it is catalyzed by acid or enzyme. When a fatty acid is treated with water, it is catalyzed by acid or enzyme. There comes the formation of there comes the conversion of fatty acid into respective glycerol and respective fatty acid, this one. Saponification with the NaOH, when it is treated, or triglyceride, when it is treated with NaOH, then there is a kind of a soap formation. It happens in that step. Glycerol is here, you can see, and there is the formation of sodium fatty acid. This is kind of so. Saponification reaction produce soaps. Hydrophilic and hydrophobic regions are produced. This is again the hydrophilic, this again hydrophobic, and hydrophobic dissolved in oils. Hydrophilic stays on the surface. Insoluble salts form with hard water. However, when there is hard water in insoluble salts, they settle at the surface with the sodium. They are produced. This is kind of sodium stearate. It is 18 carbon bearing unsaturated structure, and the sodium dodecyl sulfate is 12 carbon bearing saturated sodium salt. Hydrogenation, when in the unsaturated fatty acid or triglyceride, when there is the addition of hydrogen, then complete, partial or complete hydrogenation is produced, and saturated fats are produced. It changes plant oil to margarine. This is the oil kind of structure, and this is the solid structure. So from oil to ghee or margarine. For that reaction, hydrogen is added through when platinum is used as catalyst. Chemical reactions, this is the overview. Triglycerides, when they are hydrolyzed, or they produce glycerol or fatty acid, and this can be obtained vice versa. Similarly, when triglycerides are reacted with NaOH, glycerol is produced along with the fatty acid salt. More saturated triglyceride is produced from unsaturated triglyceride after reaction with hydrogen in the presence of nickel, platinum, or any catalyst. Artificial fats. Fats can also be produced artificially. They are the simplest microparticles of proteins, smooth cream texture, not heat soluble, stable. They produce oil when heat is applied, and the calorific value is 1.3 calories per gram as compared to fats, that is 9 calories per gram, that is for fats. This is calorific value for proteins, and this is for fats. Olestra, polymer of sucrose and fatty acid. It is kind of artificial fat, and it is the polymer of sucrose along with fatty acids. They are not digestible. Taste and texture of fats cause loss of fat soluble vitamins and other side effects. Waxes is kind of a fat, like it must be having unsaturated hydrocarbons. Long chain fatty acids plus long chain fatty alcohols, they are the saturated fatty acids. Yes, this is the example that is having no carbon double bond myristyl palmitate, one of the structures that is used for the wax. Wax block diagram. Long chain fatty acid along with long chain alcohol, not the glycerol. Glycerol is not long chain alcohol, but to build a wax you need long chain fatty acid along with long chain alcohol. Then comes the phosphoglycerides, triglycerides. In triglyceride, this was the structure. There are 2 fatty acids, and in triglyceride there was the 3rd fatty acid down there. But in phosphoglycerides you can see phosphate along with alcohol, they are in combination with the glycerol and this 2 fatty acid structure. You see glycerol, 2 fatty acids, phosphate, and another alcohol. They produce phosphoglycerides. Phosphatidic acid. Phosphatidic acid is glycerol along with 2 fatty acids, and there is phosphoric acid addition as a third carbon of this glycerol. Alcohols used in phosphoglycerides. There are 3 types of alcohols. You see this alcohol bearing group. This structure name is choline. Then there is at one end there is acid, one end there is OH, and there is NH2. This is kind of amino acid serine. Then there comes ethanolamine, OH, NH2 at both ends, and there is ethene in the middle, ethanolamine. When these 3 different alcohols they unite or they combine with the triglycerides, different phosphoglycerides are produced, different structures are produced. First one is phosphatidylcholine. This choline along with phosphoric acid, it is introduced in the structure. This kind of phosphoglycerides are produced. This is phosphatidylserine. When there is addition of this serine amino acid. Similarly, there are other triglycerides, ceramide. You need to practice these structures on the paper, and only 1 or 2 structures will be there in the quiz. Sphingosine fatty is the combination of sphingosine. This is the sphingosine, fatty acid, phosphate, and choline, phosphoric acid and choline. This sphingomyelin found in myelin sheet under neurons. It's all about lipids. Sphingomyelin, this is the structure in detail, fatty acid having phosphate groups and choline, myotin. These are the glycolipids, lipids and carbohydrate. Here you see cerboside, that is the combination of sphingosine. Sphingosine is the structure down there. Yeah. And then fatty acids, this one, carbohydrate, this structure, they constitute cerboside. Lipid storage diseases originated from abnormal metabolism and accumulation of glycolipids and sphingomyelin. These are the different diseases of the brain, spleen, liver, and other organs. I hope this lecture will be sufficient to explore the view of the lipids and fatty acids. In the next lecture, we will discuss in detail the fatty acids. Till now, you can overview these 2 lectures for a detailed content of lipids. Thank you.